ASRB NET 2021. So plant pathology question paper. These are memory based question paper because while I was writing in 2021, me and along with my friends who came to write the exam, so we remembered and collected all the net questions together and compiled in a single format. So my name is NH Shankar Reddy and I am currently working as assistant professor in Kasar College of Agriculture Science and Applied Research, Bestayu. It's near Anantapur, Andhra Pradesh. So let's see. So the first question is contagium vivum fluidum. So this statement is related to which scientist? They are asking. They gave us a statement like contagium vivum fluidum. This statement is related to M.J. Bijernek. So M.J. Bijernek is not. So he's also considered as a father of plant virology because he is the first one who coined the term virus. He also coined the term virus, and that tobacco mosaic liquid, that infectious liquid, he named that liquid as contagium vivum fluidum. So this statement is actually related to Bijernek. He is also considered as father of plant virology, and he is also coined the term virus in the sense poison or venom liquid. So the next one is who is considered as the reconstructor of mycology or Linnaeus of mycology. So E.M. Fries is considered as a Linnaeus of mycology. E.M. Fries is also considered as a reconstructor of mycology or Linnaeus of mycology. So this E.M. Fries and one more scientist C.H. Persoon. So these two scientists are the they are the students of Linnaeus. Students of Linnaeus, careless one Linnaeus, where we are binomial nomenclature and all the things that was given by Linnaeus only. This E.M. Fries and C.H. Persoon are the students of Linnaeus, and where later the students become great contributors to the mycology and as well as plant pathology. So E.M. Fries is considered as a reconstructor of mycology, or it is also considered Linnaeus of mycology. So the next question is scientific principles of plant disease management was given by. So scientific principles for plant management was given by. I have no idea because what all the options that was given was actually forgot. But if anybody knows, please comment below. And when it comes to the fourth question, so the revolutionary germ theory of diseases was first initiated by. So it was first initiated by Louis Pasteur. Louis Pasteur is a very famous scientist, microbiologist, and is also considered as father of modern microbiology. And the revolutionary germ theory was first initiated by Louis Pasteur only. Before that, many scientists like Spallanzani and other scientists was they can't they gave the proof, the programmed proof that actually diseases are came actually from the pathogens, or not directly from the pathogens, but what their statement is the diseases or the infections come from non-biotic agents. So Aristotle used to say that infections, all this decaying and all those things, they thought that which originated from non-animate agents or inanimate agents. So later they scientists were like Louis Pasteur or maybe Antoine de Berry and Robert Koch. These are all laying very strong foundation for germ theory of disease, which means if there is any disease is causing, there might be a pathogen responsible. There might be a pathogen behind that disease. That is called germ theory. So this was first initiated by Louis Pasteur only. Okay, and let comes to the next question, fifth question. So the pioneer for the fungal virulence gene cloning or isolation and characterization. So the fungal virulence gene was first cloned by DeWitt. When it comes to the bacterial virulent gene, it was first cloned by Staskowitz. So fungal virulent gene was first cloned by DeWitt. Okay, and when it comes to the bacterial virulent gene, it is Staskowitz. The year is I think 1984 maybe. When it comes to the DeWitt, that is the fungal virulent gene was first isolated by DeWitt in the year of 1991. I'm not sure, maybe 91 or 94. I thought that's maybe 91. So please check once again before making this statement is right. So the next one is nucleic acid is the infectious material of plant virus, is given by Geiger and Scram. Geiger and Scram. It's a very important. It's a very important. So before that, before Geiger and Scram, many scientists thought that protein coat is the infectious material. I will tell you one more example. We know very well W.M. Stanley. He's a very famous scientist, renowned for the crystallization of tobacco mosaic virus. For that, he also got Nobel Prize in 1935. So here, W.M. Stanley also thought that protein coat is the infectious material of plant virus. So later, Geiger and Scram in the year of 1956, they come up with a program to prove that nucleic acid is the infectious material of plant virus. protein coat only protects the nucleic acid and it has a no role in disease spread or disease infection. So it's a very important question, because I have seen the same question 2-3 times, because so far I wrote net exam 4 times. I have seen this question, I think it's around 2 to 3 times I have seen this question, not only in net but also in various other exams also we have seen this question. So it will be very important one. So when it comes to next one, seventh one, the phenomena of parasexuality was first observed by Ponticarvo and Roper in the fungus. Which fungus that they are observed that they are asking, which the fungus is Aspergillus nidulans. Aspergillus nidulans. It's a very important question. They can also ask questions like this in previous net question papers. I have seen that they asked the parasexuality phenomena was discovered by, so we know very well. So it was discovered by Ponticarvo and Roper. Ponticarvo and Roper. So they discovered the phenomena of parasexuality in 1952 in the fungus Aspergillus nidulans. Let's come to what is parasexuality. So when it comes to the parasexuality, plasmogamy, karyogamy, meiosis not occurs in a regular stages in a specified manner at a specific point of times. Actually, we know if you see the sexual reproduction of fungi, plasmogamy, karyogamy, meiosis occurs in a regular stage, right? Regular stage. So in plasmogamy, there are like gametogamy, gametangial copulation, gametangial contact, spermatization, and somatogamy. So many things are there. So, but when you see the order of sexual reproduction, plasmogamy, karyogamy, and meiosis occurs in a regular stage, like a point 1, 2, 3, something like that. But when it comes to the phenomenon of parasexuality, it won't happen in a regular stage at a specific point at a specified manner. So this is called parasexuality, and it was first discovered by Ponticarvo and Roper in Aspergillus nidulans in 1952. Year is also important. I have seen the year many times. So the next one is eighth one, the complex disease cycle of cedar apple rust alternating between two host plants was first delineated by. So it was I think in fact I too don't know the answer, but I just remembered the options and I came back to room and I just checked and I got the answers that Andres and Sando Orested. So these two are the one who alternated the differences between the host plants and the cedar apple rust. That was delineated by Andres and Sando Orested. So the next one is Ralstonia solanacearum race 3 biovar 2 asymptomatically present in which host? They have given some hosts. I was completely out of it. I have no idea. Please, if you guys know the answer, please comment below. So the next one is stalk rot of corn. So stalk rot of corn is caused by Irvinia dissolvens. They have given many options, but the exact answer is Irvinia dissolvens. I thought that some other answer, but when I came back and checked the answer, I came to know that Irvinia dissolvens is the pathogen which is responsible for stalk rot of corn. The next one is agar-agar, which is used as a common solidifying agent in microbiology and molecular labs, which chemically contains. So previously, if you see 2018 1 and 18 2 as well as 14 question papers, the question will be like this: agar-agar is isolated from. So we know very well Gelidium species, and as well as one more species is also there, Gracilaria species. So agar-agar is actually isolated from red algae or red seaweed, that is Gracilaria, as well, and Gelidium species. Gelidium is a very important one. If you see the previous question paper, the question will be like this. Now they slightly twisted the question. So agar-agar is actually commonly solidifying agent. We know very well that, and it is chemically contains. So agarose and agaropectin are the two compounds it is chemically composed of. So the question will be, they can also ask like this: what are all the units or the composing units of agar-agar? So it is a D-galactropyranose units. It is actually agar-agar is composed of D-galactropyranose units. There might be a chance, so they can also ask questions like this. But previously agar-agar is isolated from such kind of very simple questions. We know very well that agar-agar is isolated from red seaweed or red sea algae, that is Gracilaria species, and as well as Gelidium species. Now the question is, it is chemically contains agarose and agaropectin. These are all the two chemical compounds which agar is composed of or chemically contains. So it is a very solidifying agent, agar-agar. So it is actually suggested by Ernie she was she was a wife of Tiselius, where he was working in the laboratory of Robert Koch, and before that gelatin was using as a solidifying agent. So few bacteria may digest the gelatin because the gelatin is not a stable compound or not a stable solidifying agent. So after the invention of this agar agar, it act as it is a very stable solidifying agent. So not only it cannot be digested by any other bacteria or maybe fungi. So after the development or discovery of this agar agar, so everyone start to use this agar agar as a solidifying agent, and it melts at 98 degrees centigrade and it sets at 45 degrees centigrade. It is the best solidifying agent that can be used in any microbiology laboratory, and it is isolated from gelidium species and gracilaria species. It is contains chemically contains agarose and agaropectin. So when you see the bonds, as I told you, that D-galactronopyranose units, where it is composed of. And when it comes to the next one, Ralstonia solanacearum, it is a bacterial pathogen, is a non-pathogenic on members of Poaceae. So in Poaceae, what the options given is like fruits has been given and some other vegetables has been given. In fruits and especially vegetables, we can clearly see that Ralstonia solanacearum can easily infect fruits and as well as vegetables. But when it comes to the Poaceae family, I think there is no Ralstonia solanacearum. I might be wrong also. Maybe as far of my experience, it's a Ralstonia solanacearum. There is no such kind of infection that can be observed in Poaceae family, members of Poaceae family. Please correct me if I am wrong. And the next one is the race and disease of Ralstonia solanacearum that is not reported in India from. I have no idea what exactly it is. Please comment below if you guys know the answer. And the next one is the Rossetti disease, that is RRD disease. Emera virus is caused by. I haven't remembered the options also. Please comment below if you know the answer. And the next one is also I have no idea, that trichomoviruses are the members of potiviruses and is actually transmitted by. I think the options are aphids, maybe leaf hopper, something like that. The general options they have given. So please let me come to know if you guys know that trichomovirus are the members of potiviruses. It is transmitted by. So I think maybe aphids. I'm not sure. I repeat, I'm not sure. Maybe. And effector triggered immunity, that is ETA is it is a monogenic or qualitative, monogenic and qualitative. So they have given like monogenic or polygenic or something like that. They have given what I thought is it's maybe monogenic. And the next one is skin water soaked, comma light colored, comma greasy translucent, comma blistered with a mass of gas and decomposing cell is a characteristics of. So I have typed the same thing in the internet. What I found is Irvinia aroidae, and the same option is also there in the exam. So I thought this Irvinia aroidae is the right suitable answer for this water soaked in a light colored greasy translucent blighted with a mass of gas and decomposed cell is the characteristics of Irvinia aroidae. And the next one is original origin of stripe rust or yellow rust of wheat is originated from. So where it is actually originated is I have no idea, because I have seen some texts like maybe it's maybe originating from USA or maybe Mexico, something like that, but I'm not sure. Please let me come to know if you guys know the answer, and please comment below. And the action of phytoalexins, but generally what I know is the phytoalexins are like it's normally ruptures the plasma membrane or inhibition of production of fungal enzymes, and it also inhibits the spore germination and germ tube elongation. But they haven't given, there is no such kind of options I have seen in the exams. But the general knowledge what I have about the phytotoxins is rupturing of plasma membrane and inhibition of fungal enzymes, and it also inhibits the spore germination as well as germ tube elongation. But the options were not there in the exam. I think it might be something else. And the next one is Xanthomonas bacterial wilt of banana, which is caused by Xanthomonas campestris pathover malvacearum, was the first reported from. In exams, I don't know the answer. Later, I come to know it was the first actually originated and infected on banana in Ethiopia. So this was the first reported in Ethiopia, but. There might be some other location also. If it is not right, please let me come to know. So the next one is pathover aviar gene product. pathovar aviar gene product is also called as, I have no idea, because what I know is pathovar virulence genes. These are the concept was first given by like H.H. Flor in 1950, and the name of virulent gene, and H.H. Flor also coined the term virulent gene. But what they are asking is pathogen aviar gene product is also called as, it is also called as, I have no idea. So please let me come to know if you know. And the next one is outcome of ETA in plant. What is the outcome of ETA in plant? Effect or triggered immunity? I have no idea. And which of the following viruses are aphid transmitted? So potato virus, why? So it is a aphid transmitted virus, and maybe other options also will be there. But what I know is the potato virus, why it is aphid transmitted disease. And among the Gemini viridae, next question, among the Gemini which of the following group is aphid transmitted? We know that Gemini means twin particles. So Gemini viridae is a twin particular group which consists of genome, single stranded DNA. But what they are telling you, which of the following Gemini viridae group is aphid transmitted? I have no idea. Please let me come to know if you know. And the next question is the members of Potiviridae family is mostly transmitted by. This is very clear that most of the Potiviridae family is transmitted by the vector species aphids. So aphids are the major agents which are involved in the transmission of Potiviridae. Not only Potiviridae, if you see most of the plant viral diseases, around 50 to 60% of the plant viral diseases are transmitted by aphids. If you see the 60 to 70% of the plant viral disease alone are transmitted by aphids and leafhoppers. These aphids and leafhoppers are the main culprits which are transmitting at least 60 to 70% of the almost all plant viral diseases. So what I know is, as per of 2011 scenario, around 2000 plant viruses has been reported. Among the 2000 reported plant viruses, 60 to 70% are alone transmitted by aphids and as well as leafhoppers, and plant hoppers also too, but mostly aphids and leafhoppers. And the next one is plant defense suppression in plant pathogenic bacteria of Xanthomonas is true. I have no idea. What they are trying to ask is, I didn't even lighted in my mind. But please let me come to know if you know the answer. And which of the following plant pathogen enters into the plant cells through lenticels? So commonly, potato scab normally enters into the plant cells through lenticels, but there is no such option was there in exam. That's why I mentioned that, but no option here. So, but potato common scab can enter into the plant through lenticels, but the options that was given is completely, striking, like very new pathogens that they have given. So I have no idea. If anybody wrote and attempted the same 2021 question paper, if you know the answer, please let me come to know. And who introduces the cultivar mixture in the management of plant diseases? I have no idea. The next one is general review of applied mycology was started by. I have no idea. Ability of a bacteria to propel themselves over a solid surface by using type 4 villi. So normally type 4 villi can be observed in Agrobacterium tumefaciens. It's a very important one. Type 4 kind of villi can be observed in Agrobacterium tumefaciens type. So the ability of a bacteria to propel themselves over the solid surface by using type 4 villi. I don't know whether they asked the bacteria name or their what kind of options that were there. Actually forgot, it's been so long, right? So please let me come to know if you guys know. So the next one is the type 4 pili is a type of motility. As I told you just now itself, the type 4 is pili can be mainly observed in Agrobacterium tumefaciens. So when it comes to the Agrobacterium tumefaciens, the type of motility of Agrobacterium tumefaciens is swimming motility. Normally swimming kind of motility can be seen in Agrobacterium tumefaciens, but they are specifically asking type 4 villi. Maybe some other bacteria also may fall under type 4 villi, but what I know is Agrobacterium tumefaciens contains type 4 villi. Agrobacterium tumefaciens can swim through swimming motility. So that is the motility for Agrobacterium tumefaciens. I might be also wrong. Please let me come to know. And the next one is the bacterial fruit blotch of cucurbits is caused by. So bacterial fruit blotch of cucurbits is caused by Acidovorax citrulli. It's a confirm one. And terminal necrosis and wilting of papaya is caused by. I have no idea about this. And the next one is in which non-persistent transmitted virus that capsid only involved in the transmission during insect vector. So here there is might be a particular virus that where capsid is involved in transmission of insect vector. I have no idea. Please let me come to know. And which plant virus infects virus infected infects a transmitted embryo. So which plant virus infects transmitted embryo? So bean common mosaic virus, it also spread through embryo also. So plant pathology was the first university course was started in which university? It was a little bit confusing for me because I have studied in a few books like Harvard University, and some other books also it mentioned some other university, but in internet it seems to be like Oxford University, but I have no idea maybe where it is exactly actually started. But what I studied is Oxford University in very long back, around 1870s and 1880s or maybe 1890s. So please let me come to know. And the oldest plant pathology society, it is a very crystal clear Netherlands Society of Plant Pathology, which was started in 1891. If you see American Phytopathological Society, that was started in 1998. Japan Society of Plant Pathology, I think it's 1916. And we know very well that Indian Phytopathological Society was started in the year of 1947. 1947 under B.B. Munkur. B.B. Munkur is the man who worked behind the initiation of society in India, that is Indian Phytopathological Society. But the oldest phytopathological society is definitely Netherlands Society of Plant Pathology. So coming to the next one is zebra chip of potato is caused by Candidatus Liberibacter acidatus. It's a very 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 important one. I have seen this question in many places, not in net, but in other exams I have seen this question. So zebra chip of potato is caused by Candidatus Liberibacter acidatus. And the next one is the condition that favors the occurrence of epidemics. I have no idea what they are trying to ask. And the, if you see the 2021 question paper, the options also like too much confusing. Maybe confusing for me, it might be not for you guys maybe, but the options are like a little bit tricky and a little bit confusing because we were in that much pressure on the time. Even though it's a very easy question, we might get up it twisted our mind, like so hope you understand the situation in the exam. So when it comes to the next one, epidemics generally occurred due to, so sexual spores, asexual spores, sclerotia and chlamydospore, these are all the option. So chlamydospores are the resting spore, there is no chance. Sclerotia is also a soil borne spore, there is also no chance. So the, I strongly believe, not strongly believe, the answer is sexual spores, because the rate of reproduction is continuously high. When it comes to the sexual spore, the rate of reproduction, inoculum load, and continuously the spores will multiply, continuously multiply. So if the spores are multiplying continuously, epidemics will be very severe. So most of the epidemics occurs due to airborne pathogens. If you see airborne pathogens, because the rate of reproduction, the rate of reproduction is very fast, so that sexual spores or sexual reproduction, which plays a major role in epidemics. So I think epidemics is also mainly due to the sexual spores. So let me come to know if I know if it is wrong. The next one is in fungi, how many nucleotides are present in 5 to 6 MB? It's around 4,000 to 6,000 genomes are there. What I feel is around 4,000 to 6,000 genomes in 5 to 6 MB. Maybe it's wrong also. Please let me come to know if it is wrong. And the next one is number of proteins can be coded from fungal genomes. I don't know what I studied and I have seen in many areas is around 20,000 genes. Around 20,000 genes. Number of proteins that can be coded by a fungal genome, around 20,000. I'm not sure, this might be also wrong. I'm just telling. I've seen somewhere else. I'm just telling here. So it might be also wrong. Please correct me if I'm wrong. And the next one is 43 examples of amphimobile fungicide. So the options given is metaloxyl is the most suitable one, because phosphatile aluminum is also the best example of amphimobile fungicide. Amphimobile in the sense the fungicides can go acropetal and as well as basipetal, in the sense upward direction, and the action will be downward direction, both direction it will be there. So the best example is metaloxyl and phosphatile aluminum. In options there is no phosphatile aluminum, definitely it will be metaloxyl only. So the next one is what is the initial name of fire blight of pathogen that was named by T.J. Burrill in 1882. See, the thing is, what we are studying, that fire blight of apple is caused by Irvinia amylovora, right? So Irvinia, that genus was first given by Smith in 1920. So to honor the Smith, a new genus was created, that is Irvinia, because E.F. Smith is considered as the father of plant bacteriology, right? Irwin Chargaff Smith, E.F. Irwin Smith. So that the name Irwin starting, how his name is starting. So to honor the Smith, because Smith is considered as father of plant bacteriology, and he worked almost for 20 to 30 years on only plant pathogenic bacteria, because he made really a significant contribution in the field of plant bacteriology. That's why he renowned as the father of plant bacteriology. So but T.J. Burrill was the first one who identified the bacteria, who gave the first proof that bacteria can also cause plant diseases. So the first plant bacterial disease, fire blight of apple, the name that was given by T.J. Burrill is Micrococcus amylovirus. Later, the Micrococcus amylovirus name changed to Irvinia amylovora. But the original name that was given by Micrococcus amylovirus, and the year is 1882. It's not 1878. The many books mentioned it is 1878. What my research I have seen in many books and many standard things, that 1882 only in the same T.J. Burrill come to a final proof that plant bacteria can cause a disease. That is the first plant pathogenic bacteria to be identified by T.J. Burrill in the 1882, that is fire blight of apple, which is caused by Irvinia amylovora. The initial name of Irvinia amylovora is Micrococcus amylovirus. So the next one is, what is the first plant pathogenic bacterium known to cause trouble in plants? So it is Irvinia amylovora. As I told you, that the first plant pathogenic bacteria that was discovered by Irvinia amylovora only. Okay, and the next one is dwarf bunt of wheat is caused by. So dwarf bunt of wheat is caused by Tilicia controversa. Then next one is gene for gene hypothesis in 1946 was first given by H.H. Flor. We know very well. And let me talk something about this gene for gene hypothesis, which was first given by H.H. Flor, Henry Harold Flor in 1946. And he was first working on linseed rust, Lena mysorens, Lena mixotatinum are the scientific names. So if you see this gene for gene hypothesis, for every resistant gene in the host, there must be a corresponding and a virulence gene in the pathogen. That is a gene for gene hypothesis. For example, if there is a virulent gene, if there is a strong gene which will be there in the plant can produce corresponding virulent gene to tackle that, because it need to cause infection, right? If there is no virulent gene or strong gene that tackles plant, the pathogen cannot able to cause infection, right? Even though the pathogen come up with a new gene, again after a few days the plant also come up with some other gene. That's why they used to say, for every resistant gene in the host, there must be a corresponding virulence gene in the pathogen, and vice versa. At ends they add vice versa. It happens in the plant, in the meantime pathogen also. So this was first given by H.H. Flor. And 48, gene for gene hypothesis can exist in, it's a gene for gene hypothesis can exist in continuous pathosystem. I have no idea what is continuous pathosystem, but many of my friends and other people told that it can exist in continuous pathosystem only. And the next one is virulence hypothesis was proposed by, what I know is virulence hypothesis, that virulence on the concept was which is given by H.H. Flor only. But they're asking virulence hypothesis. If you see the options, there is no H.H. Flor. And later I came to know Gopalan et al. In 1996, they was given the first virulence hypothesis. But I might be wrong also, because it's been very long. So I have almost forward the options that what they have given. But if you see the research articles in other areas, what I found is Gopalan et al. In 1996 were the first one who came up with the virulence hypothesis, and they found that artificial expression of Pseudomonas syringae, that is AVRB in Arabidopsis. So that was the first progress proof of virulence hypothesis that was given by Gopalan et al. In 1996. But I'm not sure whether this Gopalan option was there in that exam or not. So the next one is the Burgundy mixture composition. We know very well Bordeaux mixture, which was given by, developed by Melody. It is also called as serendipity discovery. I'm talking about Bordeaux mixture, not Burgundy mixture. Okay. So the composition of Bordeaux mixture is 1:1:100. 1 kilogram of copper sulfate, 1 kilogram of hydrated lime or lime, 100 liters of water. That can give the exact composition of Bordeaux mixture. It is also called as holy water. Holy water. I can also say it as fungicide of the century, because there is no such fungicide has been developed. Still it's been almost if you see 140 years since the discovery, still the scientists are sorry, not only scientists but also the farmers are recommending and using this bodo mixture for the control of various diseases, for the various diseases, and also cost effective, right? We can easily prepare it. When it comes to burgundy mixture was developed, as I told you, that bodo mixture was developed by Millardet in 1882, in between 1885, because he come up with the final proof in 1885 only, along with one more farmer. And when it comes to the burgundy mixture, so the burgundy mixture was developed by Mason, Masson. So the burgundy mixture was developed by Mason, Masson, in 1897. Sorry, maybe I'm not sure, the year was not right, maybe year is different, maybe I thought that it's 1897 only, I'm not sure, please go through you guys. But burgundy mixture, the composition of burgundy mixture is copper sulfate, sodium carbonate and water, the same 1:1:100 composition only. But instead of lime in bodo mixture, burgundy mixture is replaced with sodium carbonate. Let me come again. 1:1:100, 1 kilogram of copper sulfate, 1 kilogram of lime, 100 liters of water is the composition for bodo mixture. So when it comes to the burgundy mixture, 1 kilogram of copper sulfate and 1 kilogram of sodium carbonate and 100 liters of water. Instead of lime in bodo mixture, burgundy mixture replaced sodium carbonate. That's it. That is the burgundy mixture that can be recommended for the management of various diseases. And the next one is fungicide with QOI, that is quinone outside inhibitor. Inhibitors are effective against the management. I have no idea. Quinol fungicides are effective against. And the mode of action of strobilurin. So the strobilurins are the new generation fungicides which actually isolated from a mushroom called Strobilurus tenacellus. Strobilurus tenacellus, it is the mushroom where the most of the new generation strobilurin group of fungicides are isolated from. Generally the mode of action is, that is inhibition of mitochondrial respiration. Generally I am telling, because the mode of action is very long, but simply I am telling that is the generally it's a mitochondrial inhibition. But in exam there is no such kind of option I have seen, so it might be maybe something else also. And please let me come to know if you guys know. And the next one is the bitter pit of apple is called by, is it is a calcium deficiency. It is a very standard question, and I have seen in many areas. And the next one is why staining is done for phloem cells for phytoplasma identification. I have no idea. Please let me come to know. I think scientists or maybe someone, if someone are seeing this video, please let me come to know. There might be some reason definitely behind. So 55. Enzymes used in amplification of a template in loop mediated isothermal amplification, that is BST polymerase. It's a very standard one, BST polymerase. So we'll see what is the LAMP, loop mediated isothermal amplification. It is a amplification technique that was developed by Notomi in 2000. Notomi is a scientist who biochemist who developed the technique called LAMP. It's like a PCR only. Okay, in PCR there might be three temperature like denaturation, annealing and extension. We followed three temperature, 92, 95, 55 to 65, and again extension 72. So when it comes to LAMP, we don't need to go with these three kinds of temperatures like 55, 90 something blah blah blah, and we can maintain only one standard temperature, that is 65 degrees centigrade. 65 degrees centigrade, all the reactions will happen there itself. But we are using here like an amplification template that is called BST polymerase. It's a standard one for LAMP, loop mediated isothermal amplification. When it comes to the next one, physiological effect of rice with yellowing of young leaves is due to nitrogen deficiency. Okay, and next one is Xanthomonas 21. It is a resistant variety. Okay, originally isolated from Oryza langistaminata, but the options was Oryza glaberrima, Oryza sativa, Oryza langistaminata. But what I thought that it's a Oryza glaberrima, something like that. Maybe the chief source or origin source for Xanthomonas 21, that is a resistant race. But later I come to know that Oryza langistaminata is the chief source where Xanthomonas 21 is isolated. And the next one is holomorph. Holomorph means sexual reproduction and asexual, like anamorph and teleomorph combinedly called as holomorph. But there is no such kind of options I have seen at the time. But in holomorph, one sexual cycles and many asexual cycles can be seen in holomorph. It's a combination of sexual and asexual, but one small change, one sexual cycle and many asexual cycles can be observed as holomorph. And the next one is phytoantispensor. We know very well phytoalexins and phytoantispens are more or less similar only, but the options that what they are given is absolutely no idea. And what my knowledge about this phytoantispens are, they are low molecular weight compounds which are produced against the microorganisms, and phytoalexins also more or less similar, right? So when it comes to the next one. Uh, which agency is chiefly governing body for the plant virologist for classification of plant viruses? It's a very common one, ICTV, International Committee on Taxonomy of Virus. Before this ICTV set up, actually set in 1973. Before setting up this ICTV is called as ICNV, International Committee on Nomenclature of Virus, which was set up in 1966. So scientists came up with a new idea. Why don't we change this NV to ICTV, that is International Committee on Taxonomy of Virus. They came up with the idea of ICTV in the 1971, and they formulated a rule and they established a new body or governing body which is mainly used for classification of plant viruses, that is ICTV in 1973. Okay, here is also important. It was asked in many areas. 1973. Okay, 1973 ICTV. If it is ICNV, it's 1966. Okay, the same ICNV's name changed or renamed to ICTV, that is International Committee on Taxonomy of Virus. It is a chief governing body for the nomenclature of plant viruses. When it comes to next one is edema represents. Edema represents. Normally, edema means if over intake of water, excess intake of water that the plants are taking, it ruptures and it leads to the water soaked patches that can be observed on the leaves or maybe some other areas. So that is what I know about edema. But there is no such kind of option that I have seen. And the next one is the replication of Avicenna viridae occurs. It's a chloroplast. It's a very common and important one. Replication of Avicenna viridae happens in chloroplast. I think Pospi viridae happens in nucleus or maybe mitochondria, something else. I'm not sure. I'm not sure. But Avicenna viridae actually happens in chloroplast. And the next one is 63. How many number of oospores in a gametangial contact can be observed? That is only one oospore. Okay. And coming to the 64, correct statement related to Mycota, they gave very big statement. I haven't remembered it, but please let me come to know if anybody wrote or remembered this 2021. And next one is which virus is responsible for terminal necrosis and wilting? So tobacco ring spot virus is responsible for terminal necrosis as well as wilting. So the 66 interplot interface of a model for planting field experiments with aerially disseminated pathogen was developed by, it was first developed by R.E. Pisor and E.M. Fry were the first one who actually interplotted this model, and aerial dissemination was first developed by. And the next 67, virgins which protects tDNA, from the degradation of nuclease gene transfer into the plants. I have no idea what about this. And when it comes to the next one, 68, which bacterial pathogen, that is taxoflavin, is virulent for? It's a Burkholderia glume. Other options also I have seen like Ralston and other things, but taxoflavin acts as a virulent, especially virulent factor for this Burkholderia glume. Okay. And which of the following example is aluminum toxicity? I have no idea. And the next one is killer proteins like double-stranded RNA can be observed in which fungal pathogen? See, these killer proteins I have studied in neurospore also killer pathogens can be seen. In Saccharomyces cerevisiae also killer pathogens can be seen. But they are specifically asking killer protein double-stranded RNA can be observed in which of the fungal pathogens? They are specifically asking the fungal pathogen. I have no idea. Please let me come to know if you guys know. So the next one is world's first systemic fungicide, carboxin and oxycarboxin, was first discovered by. We know very well, it's a very very important. And the last two previous net question papers also I have seen this question, that is Van Schemling and Marshall Kulka. This carboxin and oxycarboxin, they were oxazine group of fungicide. They are actually first discovered by Van Schemling and Marshall Kulka in 1966. So the trade name of oxycarboxin is plant wax, whereas the trade name of carboxin is Vitavax. So it's important also. Oxycarboxin recommended for rust, whereas carboxin is recommended for smuts. Okay, it was first discovered by, it is the first systemic fungicide and discovered by Van Schemling and Marshall Kulka. And the next one is 72. First avirulent gene is CF9, fungal cladosporium fulvum. CF9, it is the first fungal avirulent gene. First fungal avirulent gene is cladosporium fulvum. And which pathogen affects unripened banana during early stages and it is continuous until storage? I have no idea. Maybe coltotrichum. I'm not sure. Maybe because coltotrichum may be observed in ripened stages only, and it also telling it's continuous storage. But I was a little bit doubt on this. Maybe coltotrichum and may not be also. Please let me come to know if you guys know the answer. And the next one is general wavelength of light required for the sporulation of fungi. No idea. Coconut tinangaja, coconut kadang kadang, and coconut tinangaja both are viroids. So if you see the coconut kadang kadang virus, telephone pole symptoms can be seen. When it comes to the coconut tinangaja, that infected tree look like a bending, S-shaped bending can be seen in tinangaja. Both are viroids only. Okay. So the dominant origin confer resistance to, I have no idea. The next one is the Phytoplasma is sensitive to tetracycline as well as heat. They have given two options: tetracycline, heat, tetracycline and heat, none of the above, something like that. But Phytoplasma is sensitive to tetracycline as well as heat also. And the next one is 78 beta 1,3 glucanase is a polymer formed in a defense response of, I have no idea. And the small hair-like structure that is originated from the basis of conidia, that is called, the basis of conidia is called a denticle. That basis of conidia is denticle. I thought that it's a blepharoblast, but later I come to know it's a denticle. Okay, small hair-like structure that actually originated from the base of the conidia. That the base of the conidia is called a denticle. Okay, vertifolia effect. So we know very well the horizontal resistance, vertical resistance, vertifolia effect. This all concept was given by father of plant disease epidemiology, that is J.E. van der Planck. So what is vertifolia effect? Loss of horizontal resistance while breeding for vertical resistance, because two resistance cannot be put in a single plant, so that one will be lost. When it comes to the vertifolia effect, horizontal resistance will be present, loss of horizontal resistance due to the presence of vertical resistance. So the next one is which of the following is a abiotic disease? No idea, because they have given many options. I mean 4 options only, but it will be confusing. And plant viruses are transmitted by fungi, which is belonging to the genus Furroviruses. Okay, and the next one is first genome sequencing on the respective fields. When it comes to the plant bacteria, first plant pathogenic bacteria to be completely genome sequenced is, we know very well, Xylella fastidiosa, AJG Simpson, which actually done with the help of Brazilian consortium in the year of 2000. When it comes to plant virus, it is Xylella fastidiosa. When it comes to the human bacteria, it is Haemophilus influenza. That's why they asked the question, first genome sequencing in the respective fields. In human fields, it is Haemophilus influenza. I think it's 1996 or maybe 1998. When it comes to Xylella fastidiosa, it's definitely 2000, that was done by AJG Simpson in 2000. The next one is Piper mild mosaic virus is transmitted through a viroid. I have no idea. And then peanut bed necrosis virus is caused by tomato spotted wilt virus. And then which disease was not reported in India so far? Fire blight of India was not reported in so far, but they have given options like that. But what I believe is fire blight of apple was reported in India in many places of Himachal Pradesh and Jammu Kashmir and major apple growing areas. Fire blight was we observed in few books also. I have seen that fire blight of apple was not reported in India, but I don't know whether it's true or not. But the options, it was there, fire blight was there. And in books also I have studied fire blight which was not reported in India, but I'm not sure. Please let me come to know if you know the answer. Systematic fungicide translocated all over the plant. Systemic fungicide are apoplastic or symplectic. I don't know what proof that they are trying to give. And the next one is fungus shows the resistant to one fungicide which is also resistant to another group of fungicides. It is generally called as cross resistance. I have no idea, but it's generally, if the one fungi is resistant to one fungicide and the same fungi is also resistant to closely related fungicide, that is called cross resistant. That is called cross resistant. But I think option was there, cross resistant, but I don't know whether the that is the exact answer or not. Please let me come to know. And the next one is the alternate blight of tomato. That is early blight of tomato, which is caused by Alternaria solani. It is also called as alternate blight of tomato. Fast, Epidemiology, Tom cost, these are all are the forecasting models that can be recommended. The option is all the above. Okay, and the next one is application of boron can be recommended for the management of black tip of mango. Black tip of mango can be caused by two. What is one is sulfur dioxide toxicity, and the second one is boron deficiency also. But in the options, like deficiency has been like boron, nitrogen, or something like that has been given. But the answer is boron deficiency. So black tip of mango can be rectified by application of boron. So the next one is the sunflower downy mildew is caused by Plasmopara hall study. The next one is which of the following plant disease transmitted. So that is which of the following bacterial disease transmitted, like they have given like black rot of cabbage, something like that they have given. Irvinia trachyphylla, we know very well Irvinia trachyphylla, which is caused by Stewart wilt of corn flea beetle, that is Chitonema pulcaria, right? So this is transmitted by corn wilt, Chitonema pulcaria. Stewart wilt of corn, that is caused by pathogen is Irvinia trachyphylla, and vascular wilt is due to the toxin, that is fusaric acid. What are all the, we know very well that fusaric wilt produce vascular discolorations like brown color discolorations or maybe some other color discoloration can be seen. So this is mainly due to the toxin fusaric acid, because fusarium produces fusaric acid. I mean other toxins also produce, when it comes to the plant it's the fusaric acid, okay, like deoxynivalenol or maybe trichothecins or fuminosins. These are all the various kinds of toxin that is produced by fusarium. But when it comes to the vascular wilt, it is related to, especially related to fusaric acid only, okay. And the 94 is hot air oven for sterilization of glass versus 160 to 180, okay. And the fungicide involved in the activation of benzimidazole. It is a very standard one, important one. Benzimidazole is the fungicide which is also help in activation of systemic acquired resistance in plants. The next one is signaling molecule that activates induced systemic resistance, that is methyl jasmonate. Ethylene and methyl jasmonate can be recommended for the initiation of signals in ISR. When it comes to SAR, benzimidazole. When it comes to ISR, methylene jasmonate and ethylene also can be used, okay. And the common name of ethylene chloride, that is phosphatyl aluminum. Methyl chloride is generally called as phosphatyl aluminum. And forcibly discharged conidia is called ballistoconidia. Ballistoconidia, forcibly discharged conidia, okay. Which microscope can be used to visualize the internal compounds of a plant pathogens without staining the sample? So there is a microscope called phase contrast microscope, can be used without staining the sample. We can see the internal structures. So by using phase contrast microscope we can see. And when it comes to the next one, 100 role of condensers in microscope. So the role of general role of condensers is light controlling the light, adjusting the light, and as well as focusing the mirror. So these are all the general use of condenser. So the next one is which virulent trace of fusarium wilt of banana can affect grass mitchell. So what I know is race 1 is completely wiped out grass mitchell variety. So I think it's maybe race 1. I'm not sure. Maybe race 1, because race 1 is very severe to grass mitchell variety. So I think it's maybe race 1. And the next one is 102. Spores are produced as a result of fragmentation is called arthrospores, or fragmented spores are also called as arthrospores or arthroconidia. Okay. And the next one is Oomycetes group of fungi cell wall is made up of cellulose and a little amount of hydroxyproline. We are going to start one more other question papers of NET and last time preparation, something like that we are also planning to publish. In that I have mentioned the cell wall compositions of various, like maybe fungi, bacteria, virus, or maybe some other things. We mentioned very clearly so that we can throw more knowledge on other PowerPoint presentation. But in generally Oomycetes cell wall is made up of cellulose and as well as a little amount of hydroxyproline, okay. 104. Turkey X disease in England is due to aflatoxin. It's a very important one. Aflatoxin. Nearly 1 lakh fowls are died because of this aflatoxin infection in 1960s. 105. The conservation of floral pots into green leaf-like structure is called as virescence. You know, and virescence as well as phyllodes both, but there is no phyllodes option. I think it seems to be virescence. Okay. And when it comes to 105, bacteria used in genetic engineering. Agrobacterium tumefaciens, it is also called as farmer's best friend, and this bacteria is especially recommended for genetic engineering, okay. And 107, which of the following fungi comes under Chromista? Albo candida only comes under Chromista. Remaining all are options that was given is like maybe Saccharomyces, not Saccharomyces, some other fungi like Ascomycetes group of fungi has been given, but only Albuga that comes under Chromista. 108, which of the following rice is a physiological disorder? They have given very depthy options. I have no idea. And which of the following fungal spore can be produced in probasidium of teleospore? That is basidiospore. Okay. In probasidium, basidiospore only produced, right? In each basidium, 4 basidiospores are produced. In each ascus, 8 ascospores are produced. It's a general one. And 110, karyogamy occurs in which rustospore? Karyogamy occurs in teleospore. That's why it's bicell, right? Remaining all rustospores are single celled, but teleospore is a double celled. Okay. Classification of phytoplasma based on, I have no idea. 112, most number of plant bacterial diseases comes under grass liquids, gram negative, because gram positive very less bacteria are BSC, bacillus, streptomyces, and cyanobacterium, sorry, corynebacterium, something like that. These are all come, very few bacteria comes under gram positive. So most of the plant pathogenic bacteria comes under or falls under this gram negative, that is grass liquids. Okay. And polycyclic disease producing a curve are called sigmoid curve. Okay. Polycyclic, monocyclic, polycyclic, polyetic. Monocyclic disease produces saturation curve, whereas polycyclic disease produces sigmoid curve or S-shaped curve. S-shaped curve, sigmoid or S-shaped curve. So ISR, induced systemic resistant activity, can be observed in, no idea. And the next one is also in long sequencing of nucleotides, which of the following technique is widely used? They have given advanced NGT, next generation sequencing NGS, PacBios, and PacBios was there, 454 Illumina, Rochers, different kinds of advanced NGS techniques that I have given. What I believe is maybe PacBios, that is the most advanced one. That technique can be recommended for a long sequencing of nucleotides, and 454 Rocher, and equipment also can be used for long-term sequencing, but there will be little confusion on this. Please let me come to know if you guys know. And EPO stands for European and Mediterranean Plant Production Organization, EPO, and FRAC, Fungicide Resistance Action Committee, and 118, which one is the internally seed-borne pathogen? Wheat loose mint is a very important pathogen. Since centuries, this question was there. Best example of internally seed-borne pathogen, that is wheat loose mint. Okay. And the next one is 119, internationally reputed national collection of plant pathogenic bacteria are under the administration of, they have given this many: ICAR, FAO, ICRISAT, and UK. It seems to be maybe UK, maybe like there was one organization was there for collection of plant pathogenic bacteria. I thought my imagination, not imagination, I thought it may be UK, I'm not sure also. Okay. And the next one is, which of the following are the root parasite? Vorabanki and the Striga are the root parasite. Cuscuta and Loranthus are the stem parasites. So Vorabanki and the Striga are the root parasite. Okay. So the next one is damp. Well characterized damp in plants. They have given like cysteamine, glucan, LPS, and flagellin. These are all the options that I have given. I have no idea. Please let me come to know if you guys know. And the next one is a single celled conidia is called Ameroconidia. And the next one is the mutation rate in is high in the following development structure. Mutation rate is high in sexual spores. That's why the sexual spores are continuously evolving, right? Sexual spores are continuously evolving. That's why the new races are producing in each and every single pathogen. Suitable environment conditions for Oomycetes fungus, like moisture, maybe 90 to 95% relative humidity with cool weather or cool climate is especially favorable and recommended for this Oomycetes. And the 125, lack of iridial stage is called demicyclic. It's also very important. Lack of iridial stage is called demicyclic. The next one is triple gene block guided cell to cell movement is present in, I have no idea. And the next one is the new class of origin included that acts majority or majorly against plant viruses. I do have no idea. 128, crop losses and disease severity can be estimated to phytopathometry. CD May, I think there was a book which was written by CD May, that is phytopathometry, I think. And crop losses and disease severity can be estimated by using this phytopathometry. And the remaining options are not reliable. That's why I thought this it might be the right answer. And 129, Phylloxera vitifolia. It is a Phylloxera root aphid. Okay. Is responsible for the introduction of, it's a very famous one, grapes downy mildew, because before grapes downy mildew, Phylloxera root aphid is a very serious problem in grapes production. That's why they replaced the rootstocks with the United States rootstocks, where the real problem starts. The grapes downy mildew, unfortunately the US rootstocks are highly susceptible to grapes downy mildew, but resistant to this Phylloxera root aphid. So the Phylloxera root aphid, because of Phylloxera root aphid only they introduced the rootstocks from USA, but Phylloxera gone over. But a new disease comes, that is the grapes downy mildew, which is caused by Plasma parviticola. Miller discovered the miraculous fungicide, that is Bodomycir, for the control of grapes downy mildew only. The next one, Center for Innate Immunity is regulated by, no idea. And 132, to avoid false positive results, diagnosis assay should be specificity. It should be very specific, right? And the next one is to avoid false negative result, the diagnostic assay should be high specificity and high sensitivity. I have no idea. Please let me come to know. These two are a little bit tricky. And the next one is RXLR effector is produced by. This effector is produced by Phytophthora infestans, and chemical stimuli required for the infection of Phytophthora infestans. I have no idea. And when PAMP present in pathogens are recognized by PRRS of plants is known as no idea. And 137, inclusion bodies on cell and viral replication or assembly takes place. So in viral replication, if it is a DNA virus, the assembly takes place in nucleus. If it is a RNA virus, the assembly takes place in cytoplasm. But what they have asked is inclusion bodies in a cell where viral replication and assembly takes place, and I have no idea about this. 128, DAPA. It is a staining technique, 4,6 diamino something was there. I'm not sure. I actually forgot. DAPA staining of phytoplasma infected tissue was done due to, I have no idea. But this DAPA technique can be recommended for the identification of phytoplasma. That I know very well. 139, mobilactin B. Mobilactin A and B are the phytotoxin which is produced by rice, which is chemically. I don't know what they are actually asking. 140, phytoplasma is sensitive to. What I know is phytoplasma is sensitive to tetracycline, but there is no such kind of option can be seen in options. The next one is azoxystrobin. Azoxystrobin can be used for the effective control of. Azoxystrobin generally used for the effective control of downy mildews and powdery mildews. But in the option, I don't know whether the option was there. But generally, this azoxystrobin can be recommended for the efficient control or effective control of grapes. Not grapes. Downy mildew and powdery mildew diseases. And the next one is polymyxa and spongospora that transmits plant viral diseases. This polymyxa and spongospora disease and potato virus X, potato virus Y, this kind of disease was transmitted by this polymyxa and spongospora, and nematodes also can be transmitted by this polymyxa and spongospora species. And 143, resistant due to ETS governed by no idea. And 144, receptor like kinase RLKS for reception of signaling are present as no idea. And 145, which of the following are correct cause of deficiency symptoms? Hollow stem of crucifers, blossom end rot and the silver leaf of spinach. These three are definitely nutrient disorders. Stubby root is a nematode, right? So I think the option is C. 1, 2, 3 are the deficiency syndromes. The last one is the nematode. Though 146, plant secreting substances that cause alteration of fungal cell, no idea. And last one is among the following which is cause for the variability virulence in bacteria, no idea. So we have tried to collect the 150 questions, but we managed 147. I think the 3 we didn't remember it. But I'm telling once again, all the answers are based on the options given in the exam, and above questions are purely memory based, because not only me, along with my friends, many friends came to write the exam, and we sat together and collected all the questions and compiled in a one format. And we also started a new website, that is www.geekyresearch.com. We can give you trainings, or maybe we can guide your career, and we can also help you in teachings of or like guiding our personality developments, or many other kinds of stuff that we can provide in our website www.geekyresearch.com. And stay geeky and stay tuned. And when it comes to, so this was the book that was published by myself recently 6 months before on June 10, 2022. So a vision into plant pathology, complete student version, volume 1 and volume 2. So this book is especially recommended for students. Don't worry about the volume, it's around 1400 pages. But rather than studying many other books, I definitely suggest to go with my book. It's not because of my book, but I am telling you that I have compiled large amount of information, and vast amount of information was compiled. Definitely it will be useful for students, and the information was given is very simple, so that every single student can understand. Even a 5th class student can also understand the information. Hope this book can serve you for not only for NET exams, but also for ARS and some other competitive, any kind of competitive exam that is related to plant pathology. Trust me, this could be like student encyclopedia. I can't say mini encyclopedia, but I could say student encyclopedia. It's a small pocket encyclopedia. Definitely this book will be helpful. If you guys want, please go through, and you can also search it in Kalyani Publishers official website. So these are all about 2021 net question paper, and we also published 2007, 9, 2007, 6, 9, 2014, 1, 14 to 18, 1, 18 to 2019. So many question papers we are already published in our YouTube channel. So if you guys want, you can go through playlist. It will be useful for you.